Transgenic animals, sperm-mediated gene transfer. We are Darío Barrios Zamora and Pablo Espesa López, and we are going to present you by this video a brief summary of the different techniques to make transgenic animals, focusing on sperm-mediated gene transfer and its advantages. First of all, we are going to make a classification based on what are the object of the transgenesis. Electroporation or electropermeabilization is a molecular biology technique in which an electrical field is applied to cells in order to increase the permeability of the cell membrane, allowing chemical drugs or DNA to be introduced into the cell. In molecular biology, the process of electroporation is used to transform bacteria, yeast, or plant protoplasts by introducing new coding DNA. If bacteria and plasmids are mixed together, the plasmids can be transferred into the bacteria after electroporation. This process is approximately 10 times as effective as chemical transformation. Liposomes are artificially prepared spherical vesicles composed of a phase lipid bilayer. The liposome can be used as a vehicle for administration of nutrients and pharmaceutical drugs or exogenous DNA into the cell. Usually, these compounds will not be able of passing through the plasma membrane, so these kind of carriers were developed in order to allow the intake of these compounds by endocytosis. Viral vectors are a tool commonly used by molecular biologists to deliver genetic material into the cells. This process can be performed inside a living organism, in vivo or in the cell culture, in vitro. Viruses have evolved specialized molecular mechanism to efficiently transport their genomes inside the cell they infect. Delivery of genes by a virus is termed transduction, and this technique takes advantage of the molecular mechanism that viruses already have to introduce foreign DNA into cells. There are many types of viral vectors used nowadays: retroviruses, lentiviruses, adenoviruses, adeno-associated viruses, and nanoengineered substances. All of them have difference in their molecular using a glass macropipette to inject a liquid substance, usually DNA, to a living cell in order to modify its genetic composition. It is usually used to introduce DNA to spermatozoids and zygotes to further development in transgenic organisms. And finally, the aim topic of this video, sperm-mediated transgenesis, a method used since 1989, where it was first time proved. It is based on the intrinsic ability of sperm cells to bind and internalize DNA molecules and to transfer them into the oocyte at fertilization that could give a transgenic zygote ending up into a transgenic organism by natural development. This technique has been demonstrated to have high efficiency, low cost, and ease of use compared with the other methods we have reviewed. Sperm cells are considered by some authors as inert cells due to the fact that they do not have the majority of the molecular biochemistry machinery that somatic cells have to replicate DNA, yeast transcription, or protein synthesis. Sperm cells morphology is also particular. It is characteristic by an extremely reduced compartment and a condensed chromatin DNA nucleus connected to a very long flagellum. This observation brings us to the conclusion that sperm cells have as unique function to be the vectors of his own DNA during fertilization process. So all of this evidence make the sperm cells perfect vectors for introducing foreign DNA into cells. In fact, they are the natural techniques that nature used to combine masculine and feminine genes. The very first evidence of foreign DNA incorporation by sperm cells was obtained when they were incubated just in a solution with DNA molecules. It was described by Brackett in 1971. Now we are going to explain how is the natural mechanism of DNA intake by sperm cells. Here we have to define three different important things. DBP proteins, or DNA binding proteins, they are located in the sperm cell membrane, and they are responsible of recognition and binding of the exogenous DNA molecules to the sperm cell membrane at the cell head in the subchromosomal region in the proximity of the equatorial area. The MHC class II, or major histocompatibility complex of class II, they are the group of genes coding DBP proteins. They are located in chromosome 6. And the CD4 molecules, which play a very important role in the process of sperm DNA interaction and internalization. We can see all of them in the picture. So the thing is that exogenous DNA molecules are recognized by the DVP proteins of the sperm cell and get attached to them. After that, CD4 proteins take part producing the internalization of the DNA molecules by interaction with the DVP proteins and the DNA molecules. This has been proved by making MHC of class II knockout mice whose sperm cell ability to bind exogenous DNA were strongly reduced. Moreover, CD4 knockout mice were completely able to bind the exogenous DNA but they could not internalize it. To further support the role of CD4 in DNA transfer, nuclear internalization of exogenous DNA is prevented in wild-type sperm cells pre-incubated with anti-CD4 monoclonal antibodies. Nevertheless, mature spermatozoa are naturally protected against the intrusion of foreign DNA molecules. In fact, another factor, inhibitory factor 1, IF1, was described by researchers as present in seminal fluids of mammals. IF1 blocks the binding of exogenous DNA to sperm cells. In presence of this inhibitor, the DVP proteins lose their ability to bind exogenous DNA. When the DNA is inside the sperm cell, it is transcribed into RNA, which then is retrotranscribed by a retrotranscriptase to cDNA, which is integrated in the sperm genome. In order to avoid the problem of IF1 factor, epididymal sperm could be taken directly from the gonads, but it was discovered that this sperm was not completely developed, so it has a lower efficiency taking up the exogenous DNA. So what finally has been done is to take the sperm just after ejaculation, as soon as possible, and submit it through repetitive washing by several centrifugations, in order to get rid of the seminal fluid and consequently the IF1 getting isolated just the sperm cells. Then they are incubated with the foreign DNA dilute in an appropriate medium, and the whole process carries on. Now we are going to tell you some improvement of this technique to increase its efficiency by combining it with other transgenesis techniques. As other techniques have been developed to increase the entry of foreign DNA inside the sperm cell, like the usage of non-polar detergents, Triton and Twin, which destabilize the plasmatic membrane of the sperm. Similar results have been reached by freezing and unfreezing the spermatic cell, but the problem with these methods are that they usually weak the cells, so that the sperm lose fertilization ability, being necessary to use in vitro fertilization afterwards. There is another method called integration mediated by enzymatic restriction, REMI. This technique uses a linear DNA coming from a plasmid previously cut by a restriction enzyme, which leaves one cohesive ends. The foreign DNA is introduced with the restriction enzyme inside the spermatic cell by different mechanisms as liposuction, using liposomes, or electroporation. The restriction enzyme cuts the viral DNA in its specific target, where the foreign DNA will be joined by cohesive ends. It is also known that spermatic cells have a problem when they are forced to catch too much foreign DNA from outside, and they lose efficiency, so they are not able to fertilize the oocyte. So in case of big pieces of foreign DNA, another technique is used, known as sperm intracytoplasmatic injection. By this technique, we can transfer big DNA fragments by microinjection, but the problem is that we have to use again micropipettes and take care of the fragility of sperm cells that then could lose fertilization ability, having to use in vitro fertilization, so we do not really take advantage of the natural vector, the sperm cell. The last strategy used to introduce DNA in a spermatic cell is using monoclonal antibodies. The antibody is a basic protein that gets attached to the DNA by ionic interactions, and then these antibodies recognize specifically the DVP proteins, so that facilitate the entry of DNA inside the cell. In conclusion, the use of sperm-like not invasive vehicles to transfer foreign DNA to oocytes in vitro fertilization has provided a new alternative for the regeneration of transgenic animals using a natural system that do not affect the cells. As we have seen in this video, this technique is very useful because it avoids most of the problem of the other transgenic methods, which are low efficiency, elevated management outlay, and last but not least, the manipulation of embryos at early stages of development, what can be dangerous, expensive, and complicated. However, with sperm-mediated transgenesis, we avoid this embryo manipulation just by making transgenic the sperm cells. So the zygote form is from the very first moment a transgenic and will give, after proper development, an adult transgenic animal. So, in conclusion, is a very effective and relatively cheap technique that takes advantage of a natural system to make transgenic animals very useful for biomedical, veterinary, and livestock application. Thank you for watching. We hope you have understood everything clearly. Transgenic animals, sperm-mediated gene